So now I'm going to switch to flu, which is a significantly different virus in that it's a negative sense segmented, but still RNA virus. And so it's negative sense, meaning that the genome's code are not directly coding for the proteins that will make up the structural and non-structural proteins of the virus. So they have to be translated or transcribed into positive sense RNA before the new viral protein production can commence. And it's segmented in that there are actually 8 separate segments encoding the entire genome, and all 8 segments have to be encapsulated in the virus for the virus to be productive and cause productive infection. So these 8 separate genome segments encoding around 11 proteins. People occasionally discover a new protein in an alternate reading frame. Not all of those are actually required, and they come and go in different virus strains. There are 2 consistently occurring non-structural proteins, NS1 and PB1F2, although PB1F2 is also dispensable in some virus strains. NS1 is always present, though. And it has 2 surface proteins, the hemagglutinin and the neuraminidase, and that's shown here in the purple and the spiky yellow. And we abbreviate these HA and NA, and this is what gives the influenza nomenclature, the HN nomenclature, its name. And so these are serotypes, so it means they're determined by their reactivities to antibodies. So the different H's and the different N's have different broad reactivities to antibodies from animals or people that are injected with these viruses. So there are 16 broad HA. There's actually a couple more now in bats, serotypes. All of them occur, except for the bat ones, in aquatic birds. A few have occurred in humans and routinely occur in humans. Right now we have a lot of H1 and H3. In the past we've had H2. And then there's jumps of these other species or strains, H5 and H7 you probably heard about. And there are 9 Ns. And so in principle, you could have 16 by 9 different serotype viruses. In practice, again, we have some restrictions on the pairings between these H's and N's and what fits in humans. All of these serotypes are always circulating in aquatic waterfowl, though, which are the definitive host of influenza viruses. Okay. As I said, the aquatic waterfowl are the definitive host of influenza viruses. They sort of sit in the center of this group of zoonotic infections that occur from the aquatic waterfowl. But as viruses jump into these hosts, not all the viruses from the aquatic waterfowl can get into all of these hosts. And when they do jump, they tend to quickly mutate and adapt to the host that they're in, and then are unable to be spread back to the aquatic waterfowl and may not spread efficiently between other hosts. So humans are quite restricted in terms of the viruses that they've been infected with. Humans and swine tend to overlap quite a bit in what they can be infected with, and they're somewhat different than, say, the strains that infect horses or dogs. And there are some strange animals on here, seals, the whale, H13N9 and whales. And so quite a lot of diversity in influenza genomics and in the species specificity of each of the strains. So like HCV, this is an enveloped virus, meaning it has a lipid membrane, and then it has these surface proteins that'll mediate entry into the cell. And the virus gets taken up in an endosome, where it then fuses with that endosome to dump its genetic contents out into the cytoplasm of the cell. Influenza replicates in the nucleus, so those genetic contents are dumped into the cytoplasm and have signals on them that quickly get them trafficked into the nucleus, where it completes most of its viral life cycle. So it has to make positive sense RNA, and then it makes negative sense genomes again to reproduce the virus. And then from that positive sense RNA, it's going to encode mRNA that can make the viral proteins, the structural proteins, that can then get packaged by the cell's own machinery to make new viruses. So the HA, one of those 2 surface proteins, is required for cell entry. So this is the surface protein that binds to the target cell and allows it to get in the cell. It binds broadly to sialic acid, so carbohydrate moieties on the cell surface. So it doesn't have a specific proteins it binds to. It binds to a specific modification that's on many, many, many different proteins. And this triggers endocytosis by the host cell. And then the HA is the protein that also fuses the two membranes together upon a pH change inside the viral endosome. So the cell's own process to try to kill the virus, which is acidifying the endosome, actually triggers the virus to create this fusion. It lets the virus know it's in the right place because the virus is starting to get acidified. And then the HA basically stabs the host membrane, tears it open, and dumps its viral contents out into the cytoplasm. For the HA to be active, to be able to perform this stabbing and fusing motion, it has to be cleaved in a specific spot, and that protease only exists in humans, generally in the respiratory epithelium. And this is what restricts influenza to be a respiratory virus, is that these proteases are stuck in the respiratory epithelium. In birds, these proteases are often in the gut, and so influenza is actually a gut infection. So in humans, people talk about the stomach flu, but the virus is never actually really productively infecting the gut epithelium because it needs this cleavage by proteases that are restricted to the respiratory epithelium. So the neuraminidase performs the opposite function. So it cleaves the virus off the cell as it's budding away. As new viruses have been made, they actually get stuck back to the cell surface because you've got those HA molecules that are going to reach back down onto the cell and grab on. And so the NA cleaves the virus back off so that it can release and spread to other cells. And so influenza has to balance the binding of HA with the cleavage activity of NA so that they're not too out of whack of each other. So NA cleaves these sialic acid moieties off the cell surface, releasing them releasing the virus from binding them and from sticking to the cell. And so we often talk about the HA and NA activities having to be matched for activity. If you have too much of one, you either stick too hard or you get cut off too quickly. So thinking about the immune mechanisms of protection again, just like HCV, we're talking about an intracellular pathogen. And so the main mechanism of protection is going to be things that can kill infected cells. So the CD8 T cell that sees pieces of the virus in the context of peptide MHC. And potentially antibodies. And so this is going to be a case where we see that antibodies can play a role that they weren't playing for hepatitis C. So antibodies here to the HA are effective at blocking viral attachment to the sialic acid receptors on the plasma membrane. And so this is why you get the flu vaccine every year. It's actually to generate HA-specific antibody. So a course of infection in a naive human or in a naive mouse, this is data actually from a mouse, you have initial, these are now days, so a much faster replication cycle than hepatitis C, which is trying to be a chronic infection. You have the initial growth of the virus. It initially comes under control from those innate immune mechanisms. So there's this initial decline of viral titer before any adaptive immunity emerges. And then by about day 5 or so, you start seeing the earliest signals from adaptive immune responses, CD8 T cells, and flu-specific antibody that eventually clear the virus by, say, day 10 to 12. This happens much, much faster if you have memory CD8 T cells. And this is assuming that you do not have sterilizing antibody at this point. So truly homologous infection can be prevented by sterilizing antibody here. So you get sterilizing immunity to flu, at least in animal models. Okay, so how does influenza evolve, and why do we need, why don't we have sterilizing immunity forever? So the main mechanism of human influenza evolution that we see routinely is what we call antigenic drift, which is this middle panel, which is that because we have a lot of antibodies to flu, we're constantly putting the virus under pressure to mutate, and it mutates quite rapidly. A small number of mutations, so this is from the 2003 Fujian H3 strain, These were two actual mutations that occurred in the HA molecule, and it basically eliminated protection, sterilizing protection from the antibodies that were in the human population. So you got the emergence of a new strain with basically just two mutations. And so you still would have CD8 T cells here that would protect you from infection, but and potentially some weakly binding antibodies that would provide some measure of protection, but eventually, essentially you had a new strain and we needed a new vaccine. The other big mechanism of protection is antigenic shift, and that's where you get the mixture of two viruses to create a new third virus that's components of both of these viruses. And this is where the segmented genome comes into play, because if you get a cell that's infected with two different viruses, they can swap these gene segments to produce a Frankenstein virus. Now, it's not always the case that all the gene segments will work together, and some gene segments are more permissive for working with each other than others. And so in this case, you had the NA, the HA, and the PD1 gene, which is part of the polymerase complex, from one virus mixed with another virus. And this creates a new third virus that is now got a different, potentially surface genotype, because it came from the bird virus here, that's working with the innards of a human virus that allows it to replicate in a human. Okay. So this is what leads to the big shifts in influenza pandemics. So we had an initial pandemic that we've all heard of, the 1918 Spanish flu, which was an H1N1 virus. And that was replaced with one of these antigenic shift events in 1957 with the H2N2 virus, which was replaced with another antigenic shift event in 1968 with the H3N2 virus. And then in the late 70s, this exact same H1N1 virus was introduced again into the human population. This is thought to be a lab release from either Russia or China via an accidental research release. And it's considered to be that because the genome of this virus in 78 was exactly the same as the genome of this virus in 57. And it had disappeared and had not been detected widely in the human population in the intervening years, which meant it had been in a freezer somewhere and had then escaped. Both of these viruses were with us until 2009, when this H1N1, which was really the grandson of the 1918 virus, was replaced by the 2009 H1N1 pandemic. And I'm going to take you through that history here, and I'm going to show you a little model that Andreas and I have worked on of this event in the last 20 minutes or so here. Okay, so here's the 1918 human H1N1 virus, all 8 genome segments, human virus. In 1918, this virus stayed in humans. And then it also jumped into North American swine, so pigs, and it also jumped into Eurasian swine. And these two populations actually stayed quite separate from each other for the next many, many decades. They also were not under a lot of immunological pressure because we kill pigs and eat them before they can really force mutations in the virus. And so these viruses actually contained proteins that were very similar to the 1918 virus all the way through till things started changing in the late 90s. Okay, so this virus in humans stayed. I told you it went extinct in 57, reemerged from a lab in 77, 78, and was with us as the seasonal H1 until 2009. Now, in the late 90s, you had this very strange event happen in the Americas somewhere where you generated this virus that we call the triple reassortant virus. This was a combination of an avian virus that donated 2 components of the polymerase complex, the North American swine virus, which donated the HA, so this H1HA, which had sort of been there since 1918, and then several structural and non-structural proteins, and then a human, weirdly, H3 virus. So 3 different viruses, a human virus, a pig virus, and a bird virus met in a pig and generated this swine virus. And all of a sudden, the American swine lineage that had been completely boring and predictable for 80 years started mutating like crazy. You started getting all of these reassortant viruses, reassortants with humans, reassortants with birds, and all on this background that we called the trig backbone for triple reassorted genome. And this virus met a Eurasian virus at some point in the mid-2000s, somewhere, we don't know where. And the Eurasian virus donated the NA and M, so the neuraminidase and the matrix protein, which is another structural protein, to this triple reassortant virus. And it donated the N1 specifically from this Eurasian swine lineage that had been boring in Europe for 80 years. And that created the pandemic H1N1. And this was a very successful virus. People sort of think of this as a dud of a virus, I think, particularly now in the context of coronavirus, because we didn't have worldwide lockdowns and not everybody died. But within 6 months, a third of the world had basically been infected with this virus, which from the virus's perspective is really, pretty darn successful. And this is, the rapid spread and incredible infection rate of this virus, I think, informed a lot of people's thinking, including my own, of what was going to happen with coronavirus if we didn't take measures. So, basically, we didn't really take any extreme measures with H1N1 because we knew we could make a vaccine very quickly, and because of some memory immunity, which I'm going to show you some evidence for in a couple slides. But still a third of the world was infected in 6 months. Very successful virus. So we have a question in the chat. When you were saying the viruses met, does that mean their respective genetic code were produced together in a cell? Exactly right. So they had to meet in the same pig, in the same cell, in the same pig. And we're pretty sure it was a pig. That's what I mean by viruses met. And then the other question in there is, does the cleaving of HA by protease serve to enable it to outcompete NA and thus favoring cell entry? No, the cleavage of HA is actually required for it to have its conformational change that allows it to perform its fusion in the cell. If it doesn't get cleaved, it can't do this flipperoo that allows it to stick its fusion peptide into the membrane. Okay, so we have this incredibly successful virus from 2 pigs, and everyone did panic in 2009. We were a little nervous that it was going to be like the 1918 pandemic, where you had incredible amounts of deaths among the elderly, incredible amounts of death among the very young, and actually incredible amounts of deaths among sort of healthy young adults. And we call this the W-shaped curve. And that's very different from the normal U-shaped curve we see in a normal year, where you have deaths among the very young and deaths among the elderly, not even very high amounts of deaths. And one thought as to what caused this W-shaped curve in 1918 is that actually middle-aged people were maybe protected by some immune memory of a similar virus. And we just don't have the ability to look back much past the late 1800s to figure out what flu viruses were circulating before them. But we assume that they were circulating. Flu didn't get invented in 1918. The other thing that was shocking about this virus was that we always assumed that you had to have a human virus as part of any antigenic shift scenario. And what we had here was actually 2 pig viruses that met. We had the Eurasian swine and the American swine, but the American swine was that weird triple reassortant virus that had a bunch of human virus components. The fact that this was quite strange, and then that was spreading so rapidly, we predicted, or not me specifically, but the medical community predicted actually significantly higher amounts of deaths than what actually occurred in the 2009 pandemic. And there was a really specific reason for that, and it's why I kept emphasizing that the North American swine had the 1918 H1 and quite stable with it all the way through to where it donated that same H1 to the triple reassortant backbone that then became the H1 in the pandemic, H1N1. And that is that people that were alive when the 1918 virus was still circulating still had antibodies. And so this shows you the power of immunological memory. This is from the New England paper that really first showed this relatively convincingly, looking at, they were trying to do a vaccine study for the new vaccines that were being made for the pandemic H1N1, the 2009 H1N1. This is looking at antibody titers in different age groups. So these are all young people, and looking at their baseline titer before vaccination. Looking here, they all have very low levels. So protection in this assay is considered to occur around a number here of 40. And all of these people, because they're very young, had very low levels of antibodies prior to vaccination. And then when they get vaccinated, they get more antibodies. If you look in in adults, also very low levels. These are now up to 64-year-olds, 40-year-old adults. But now we look in greater than 60-year-old adults. We actually have people who were protected prior to any vaccination. So they had neutralizing levels of antibody prior to seeing any vaccine. And this is even in 2007, 2008, before this virus even existed. And so what was it about these people? It's that they were born prior to 1957 when this virus was still circulating. And so they had seen this H1, they had made antibodies to it, they had made plasma cells. Those plasma cells were hanging out in their bone marrow, and they continued to make antibodies to it. And so when they saw it again, they were able to be protected. And so if you look at hospitalized cases by age group, you had one of the lowest rates of hospitalization in the elderly, which is obviously very opposite to what we're seeing with coronavirus, because we obviously have no memory of this particular coronavirus, whereas the elderly here were completely protected. If it hadn't been the case, if this H1 had been a completely novel H, let's say it was H7 or H13, then this number probably would have been horrifying. And if that had actually spread around the world and infected a third of the population, we would have seen deaths probably actually far outpacing what we're seeing with coronavirus. The young adults here actually did not do that well. And so this is something that's sort of lost in some of the news about this, the sort of memory of this virus, is that actually some of the highest rates of hospitalization were in healthy young adults, and excess deaths were actually much higher in this sort of mid-teenage group in the pandemic. They just weren't, so extreme that people considered it to be particularly bad. And you didn't have this large weight of mortality from the elderly group that would have led to sort of the sort of bad impression of this as a severe pandemic. Okay, so in the last little bit here, I'm going to show you some modeling that we did to try to understand how this virus was so fit in humans that it was able to spread around the world and infect pretty much everybody. And when you think about having had this virus, probably everyone here has antibodies now to this virus, either through vaccination or through infection, or probably both. You actually now have antibodies probably to the 1918 H1. So if sort of feel part of that long history of human exposure to these incredibly fit viruses. What we really thought was interesting was this emergence of a fit virus from two strains that were not particularly fit in humans. So you had the European swine virus that had been around for decades, and we're representing here with this swine Italy strain that we're going to do experiments with. And this triple reassortant strain, which we're going to use a North Carolina swine virus to represent, that had been around for at least 10 years before the pandemic occurred. Both of these viruses had infected plenty of humans, generally people with close contact with pigs that were infected with the viruses, and they didn't spread them to other humans. They often weren't very severe infections. They got a mild infection, they were not transmissible. There were plenty of cases, though, to initiate a pandemic if either of these viruses were fit in humans. And then these two unfit in human viruses traded two gene segments, the NA and M from the European swine strain, and all of a sudden we had a pandemic. So what was it in that mixture that led to the pandemic? And why were they so much more fit than the currently circulating seasonal H1, which is represented here by this strain? And so this is the flu nomenclature, whether it's an A or B influenza virus. If it's human, the city that it was isolated from, the number of the isolate in that particular year, and then the year. So this is a 2007 virus. It's the 59th isolate in the city of Brisbane in Australia, and it was an A strain. And then if it's an animal, the animal appears in the second spot instead of the place. So to do this, we used a totally in vitro system, and so we're ignoring now the adaptive immune response entirely. And we are looking at human epithelial cell layers, because we felt like this had to be a human experiment, because what was interesting here was the adaptation to humans. So these are 3D transwell cultures of human epithelial cells. And so there's air up here and media down here feeding these viruses. Okay, so the first thing we did was measure these viral growth kinetics of the pandemic strains, which we're measuring with 2 strains here, one from California and one from Tennessee. The American swine strain, which was that North Carolina strain, the Eurasian swine strain, which is this Italian strain, and then the Brisbane strain, which is the old seasonal H1 strain. Okay, so what you can see here is 8 hours after infection and 12 hours after infection. And what we're measuring here is infectious titer, which is what you often get in papers of viral infection. So they're measuring new viruses that can come off and can infect other cells. And you can see there's some differences here. So the pandemic strains are growing pretty well at 8 hours. Maybe the American swine's lagging. The old seasonal strain is doing relatively poorly, and the European swine strain is intermediate. By 12 hours, a lot of these differences are gone. There's some differences here. The old seasonal strain is still doing poorly. And then by 24 hours, they're basically right on top of each other. And so there are differences, but over several days, these differences weren't that big, except for potentially the old seasonal strain. If we look in swine cells, there's basically no differences. The pandemic strains, again, may be a little bit better, except for the human seasonal strain, which is very unfit in swine cells, which is probably not too surprising. Okay, so that was measuring infectious titer. And this is, Andreas hinted at this yesterday. You really got to look at what you're being given when you're modeling and what you're measuring and what's actually the relevant thing to measure. Is it really relevant to measure infectious titer if you're trying to measure the infectivity of a virus? If you want to measure the ability of that virus to transmit, sure, infectious titer is probably very important. But how well that virus gets into cells and replicates, you really want to know how many cells were infected in each of these cultures. And so we measured that directly here. And so what we're staining here is for a viral protein. This is the viral MP protein. And these are the pandemic strains again, the American swine strain, which here now looks very poor. Only 2 cells in this whole field can we see are infected. The Eurasian swine, and then the human seasonal, which now actually doesn't look so bad anymore. It actually looks much better than the American swine. And if we quantify all of that across multiple cultures, now we actually start seeing a different picture where the American swine, that triple reassortant backbone, actually has a significant lag in growth, whereas the European swine actually is doing quite well. And the American seasonal, or the old seasonal, is actually not doing that poorly. But the pandemics are definitely clearly doing the best. And look here, by 12 hours, almost 75% of the cells have been infected by the pandemic strains. And over maybe 20–50% of the cells are infected with European swine strains, whereas we had to break the graph here to show you the American swine, which are still down here only about 5%. So we measured neuraminidase activity. So that's the ability of the virus to cleave itself off the cell. And this was something a number of groups noticed. So the neuraminidase, remember, comes from the European swine strain and the pandemic strains. And all 3 strains here, the 2 pandemics and the European swine, had very high levels of neuraminidase activity, whereas the old seasonal and the American swine had very low levels. And so this might relate to the ability of the virus to spread well and create more infected cells in these in vitro cultures. And that's actually been shown by Catherine Bachemain and some other mathematical modeling papers. Okay, so what I've shown you so far is that the pandemic virus acquired this rapid growth phenotype in human cells similar to the European swine virus. It associates with both the NA and M of the European swine virus, and it transmits more efficiently in, I didn't show you this, but it transmits more efficiently in ferrets, the European swine virus. And an important thing to keep in mind, especially as you go into your modeling lives, is that titer, so infectious titer and infected cell number, can be decoupled across infections in individuals. They don't always correlate, and measuring both of them gives you independent information. So these models maybe should look familiar to you. The particular nomenclature might be a little bit different, but it's basically the models that we looked at yesterday. With a little bit of a twist. So we've got a model for an equation for uninfected cells, and uninfected cells become latently infected cells. So this is just creating a compartment so that we have cells that are infected that are not producing virus, which is an eclipse phase in viral production. And it's to try to get a little bit closer to the physiology. And so it's just sometimes in Andreas' slides, you might call these dummy compartments. And it just delays the rate at which latently infected cells then become productively infected cells. So they're now producing virus, and this is what's driving free virus. So free virus here depends on infected cells. And then cells can die, and we're actually keeping track of the dead cells. And so there's no immune response here, and we're just looking at growth kinetics. But if we did this, then these equations would predict that the European swine virus is just as fit as the pandemic strains. And it wasn't, because the European swine virus was never a pandemic. So we decided we needed to look at the immune response, but we're going to completely ignore adaptive immunity, and we're just going to ask about the innate immune response. And so again, we measured this with microarrays. Now we would do it with RNA-seq. This was in 2009. But we measured the response across 12, 16, and 24 hours in these cultures to the old seasonal, the 2 pandemic strains, the European swine virus, and the American swine virus. And what you can see is in these first 3 columns in every chart here, we're just looking, don't worry even about what the genes are at this stage. We're just looking at red means a gene comes on. We're using a very low MOI, so multiplicity of infection. So only a few cells are infected initially. It's the same MOI that we used in all of the other experiments I showed you. By 12 hours, even though the American swine virus, or the old seasonal virus, wasn't growing super rapidly, it's already turned on a lot of genes. Whereas in the 2 pandemic strains, it's almost totally black. There's almost nothing on. And the same thing in the American swine virus, there's almost nothing on. But in the European swine virus, there's quite a bit on. It looks almost like the old seasonal strain. And so we can look at just some raw data from this and just ask, what are the top genes turned on by that old seasonal strain? And they're all interferon response genes. So this is RIG-I, that OAS1 that I mentioned. IFITs are interferon response genes. MX1 is almost the top turned on interferon response gene in any system. And they're all turned on in Brisbane. They're all turned on in Italy, and they're not turned on at all. This is all noise in the pandemic strains. Despite the fact that at this time point, 12 hours, almost 50 to 75% of the cells were infected. Many more cells were infected here than in these groups. If you look at what was turned on in California and Tennessee, this is actually the top 9 genes turned on, and it's actually just statistical noise. None of these are statistically significant. The cells were completely unaware that these viruses were there, even though over half the culture has now been infected. So we called this a stealthy response. And the same thing was happening. We thought the same thing might be happening in the American swine virus, but I'm not showing it for you here because remember, the American swine virus was growing really poorly. And so we felt like it wasn't really fair to compare it head to head at a single infectious dose when it was growing so badly. So what we did is we infected across several logs of MOIs. So we added different amounts of viruses for all of the viruses across culture. And then using a high-dimensional RT-PCR, we measured a large set of interferon response genes just to see what happened. So now at every time point, we should have almost every virus matched to another virus in terms of viral burden. And so now what we're doing is plotting immune response genes, interferon response genes on the y-axis, a log scale, with a flu gene on the x-axis. So we're looking at the immune response as a function of viral burden in these cultures. And so the way you read this is the farther over you are on the x-axis, the more virus it takes to make the equivalent innate response on the y-axis. So the farther to the right and down you are, the more stealthy your response, and the farther to the left and up you are, the more noisy, the more detectable your response is and how quickly the immune response turns on in these cells. And what you can see is the purple, the Italy, is always over and to the left of the North Carolina, regardless of the dose, even if it's exactly matched in terms of viral burden to the pandemic strain, they're almost superimposable in terms of their stealthiness. And this just summarizes that across many, many genes. The North Carolina and California response were matched and were stealthy. The seasonal and Eurasian response were matched and were noisy. So then we tried to figure out what gene segment was responsible for this. And we assumed it was going to be the NS gene because that's the non-structural protein that interferes the most with type 1 interferon responses. And we individually swapped every gene to create virus hybrids between the pandemic strain and the American swine strain, and the Italian and the Eurasian swine strain. And whichever single gene swap we did, we could not recapitulate the stealthy phenotype in the Eurasian swine strain or introduce the noisy phenotype into the pandemic strain. And so it became this, and then there were none because we were making so many swaps and people were getting very frustrated in the lab. And then we had the idea that it was maybe a different Agatha Christie book, and I'm giving away the ending here, of Murder on the Orient Express, where instead of one gene doing it, it was all the genes working together doing it. And so it was we thought maybe it was the entire complex that had formed in this triple reassortant backbone that was driving this stealthy phenotype. And particularly the entire polymerase complex combined with the NS gene. And what we found was that any element of disrupting that complex by introducing the Eurasian swine genes into that complex, whether it's PB1 or PA, would actually cause an increase in the amplitude of the innate immune response here. And that came out really clearly. This is just showing that data in a different way, looking across these different gene types. Again, anytime you introduce an element of the polymerase complex from the Italian strain into the pandemic strains, you greatly increase the innate immune response that's detectable. Okay, so what I've shown you there is that the pandemic strain, the 2009 pandemic strain, was a combination of rapid plus stealthy growth, and that's what led to the pandemic. And that might imply that if that strain could take on additional HA and NAs, that it could drive future pandemics, because our innate response is not going to ever remember. It's not going to be able to remember. It has no memory, so it's not going to ever be able to learn the stealthy phenotype. And so our memory is only going to be targeted against really probably the HA and the NA in terms of antibody memory. And evidence for this is that people keep getting infected with triple reassortant viruses with H3N2 surface proteins that are getting captured, that are getting generated in swine. This still occurs to this day. Every summer this happens, probably not this summer, in state fairs in the US, and children are exposed to pigs. But it's always children, and it's always children born after the particular HAs and NAs from these H3N2 viruses were circulating. And so a lot of these viruses are in pigs from the early 90s, and children born after the early 90s are susceptible to these viruses, but everyone born before the early 90s, just like people born before the 1950s were protected against the pandemic strain, are protected against these viruses emerging. But I would imagine in about 50 or 60 years, some of these viruses, if they remain in pigs, will probably be a new pandemic. The set of genes induced by diverse viruses is largely equivalent to the first 24 hours. I didn't really show you that, but regardless of the kinetics, if you look at all the genes that get turned on across all the strains, they were actually identical. They just came on at different times. The pandemic strategy is distinct from the well-adapted human seasonal virus. And kinetic differences in the first 18 hours of infection seem to be really critical to the quality and quantity of the later response. And this echoes a little bit of what we see on a longer time scale in HCV, where those early innate responses really set up the efficiency of your adaptive response. And we think the stealthy phenotype is mediated by contributions of the entire polymerase gene complex, with potential roles for both the nucleoprotein and the NS gene segments. So now we can remodel our situation and see if we can now capture the dynamics that we actually see in culture. And we're going to introduce an immune response, which is this X here. And X can act in a couple places. It can act by reducing the number of productively infected cells, or it can act by reducing the amount of free virus that gets generated. And so, and then we had other variations on these models where we had no cell regrowth, where we did have cell regrowth, with and without the immune response, with the immune response affecting either virus production or infection rate. And so these are the 8 models. And then what we're calculating here are the goodness of fit for these 8 models using something called the KK information criteria. And we're going to go over that today. And this is one of the methods for doing model selection. And the lower the number here, the better fit for the model. And we can fit the model now with all 4 viruses. And what we see is that this one model here does somewhat significantly better than the other models, with the exception of the American swine strain, which we still have some problems fitting, but none of the models fit super well. But we can now capture the Italian Eurasian strain much better than almost any of the other models. And this is this model 6, where the immune response reduces virus production. So it works on the infected cell to lower virus production. And in the context of our culture system, we allow cells to regrow. And that fits the data the best overall. And this just shows you those goodness that fits. Okay, in the last slide here, I just want to mention, since so many of you are doing COVID modeling, and there's so many flu models, how can you adopt your COVID model to your flu model? And obviously it depends very much on your model, but I just wanted to head-to-head compare the viruses for you so you could think a little bit about how they're comparable. So they're both RNA viruses, but the coronavirus has a positive stranded genome, more like HCV. And it's a gigantic virus relative to, this is this typo, sorry, ignore this. This is not flu. Flu is 14 kb. Coronavirus is 28 to 32 kb. So it's almost twice the size, if not more than twice the size of flu, has many, many more proteins, 29 proteins versus the 10 to 14 proteins you have in the flu virus. It has a relatively similar replication cycle to everything we've been looking at. It's an envelope virus. It has to get in. It has to uncoat. It has a very complicated replication process, though, because like HCV, it has only one segment, and it makes large polypeptides that have to then be cleaved. And then it also makes small nested RNA gene segments so that it can make some of those 28 proteins without making the large polypeptide. It replicates in the cytoplasm in these large vesicles, so more like HCV than like flu, which goes into the nucleus. And one of the biggest differences, and I think this is going to be really critical for your models, is that it has a very specific receptor, and that receptor is ACE2, and that receptor is distributed throughout the body. It's not just in the respiratory epithelium, even though we think of this also as largely a respiratory virus. It's on the endothelium, it's in the gut, it's in the brain. And so a lot of what we're seeing pathology-wise is potentially a result of the wide distribution of the receptor for coronavirus. This just shows you some of those gene segments and the complexity of the coronavirus genome, which is far more complex than influenza. And again, how it makes these nested gene segments so that it can replicate and make some of these smaller proteins without having to make the big polypeptide. And this just is a nice picture, I think, of all of the many different proteins that get made by coronavirus. Again, so much more sophisticated than the influenza virus. And likely, that sophistication is allowing it to much more effectively suppress the innate immune response, even though flu already does that relatively well. And that just summarizes those differences there for you.